Okay, class 9. So here the picture there indicates you something, I guess. So if anyone can guess what is it, it's very very good for you all. So now actually, so you must be thinking what is it, no? So you all do this in the school and all, no? You have been doing it. So injecting yourself, what are you injecting there? Vaccines, no? So you should know what are vaccines and what is this process known as. So this process is actually, it is known as vaccination. Vaccination is the introduction of dead or the weakened pathogens into your body. You know what are pathogens? But they are dead or weak pathogens which are injected into your body. This technique is also called prophylaxis. This causes the body to develop resistance to a particular disease caused by the pathogens. Understanding, I guess? The germ that is introduced into the body is called a vaccine. Okay, those are dead or the weakened pathogens or the germs that has been entered through this injections into your body is known as vaccine. The serum with weakened disease causing germ is called vaccine. So vaccine when introduced causes the body to develop resistance to that particular disease. Okay, so they must be vegan enough so that you should remember the vaccines that you're putting in, the germs that are in the vaccines, okay, should be weak enough so that they do not cause any other serious illnesses. So very effective vaccines has been developed against many diseases till now, like cholera, measles, mumps, rabies, typhoid, no, and smallpox. I think so you have heard about. But the rarest disease, the deadliest disease till now, COVID-19, which is caused by coronavirus, is yet to be found. No, so vaccines have been developed in different countries and all. Okay, so under this topic, you should know there are various types of vaccines also. Here, there are mainly four types. Okay, vaccines which are developed from live bacteria or virulent germs. Okay, they are the vaccines which do not cause any diseases, but they are introduced into our body to immunize tuberculosis and many diseases like even uh, measles, yellow fever. Okay, so tuberculosis, the vaccine, BCG vaccine, no, they are developed from this live or virulent germs, but they themselves they do not cause any disease when they enter into our body. Okay, so uh, BCG, the full form of BCG is known as Bacillus of Calmette and Guerin. BCG vaccine, which is given to persons suffering from tuberculosis. They are developed from a live bacteria or a virulent germs. Second one is vaccines can be obtained from inactivated or killed bacteria or viruses. Okay, so they can be introduced into our body of infant, mainly for the person who is suffering from whooping cough. Okay, so this whooping cough is very very dangerous because it may damage, it may cause permanent brain damages also. Okay, vaccines for typhoid and polio. Okay, this TAB vaccine for typhoid and Salk vaccine for polio is also derived from killed bacteria or viruses, inactivated uh, viruses. Okay, third one is they are also vaccines that are produced from a part of virus or subunit, we call it. Okay, they can be introduced into our body. This acts as an antigen Okay, it is done with many few vaccines. The similar method is used for vaccination against hepatitis B. Then a cellular pertussis also. Okay, Haemophilus influenza type B. So all these diseases, okay, for these diseases, vaccines are developed from the part of virus itself. Okay, a unit of it is taken it. Then comes the last one is the toxoid. These are inactivated toxins, okay, secreted by the bacteria. It becomes harmless by the addition of dilute formalin, yet it is capable to produce antibodies against diseases. So these are the different types of vaccines, okay, that has been developed from different pathogens. You can see the condition live. Some are killed, some just part of it, some the toxic substance, inactive toxic substances are taken from it, toxins. No? So they are used as vaccines. Okay, here this picture, the baby is having some, taking some vaccines. No, this is a practice of making the body immune or resistant to a particular disease, and this process is known as immunization. Generally, here this technique is used for introducing dead or weakened germs into our body to develop our immunity. Remember that. Okay, so now we are going to study about different terms related with this: antitoxin, antibodies, then antiseptics, antibiotics, and all. We are going to deal with it. So here, the picture refers, okay, about antitoxins. You should know what are antitoxins, where are they used. Here, antitoxins, they provide protection against a toxin or poison that are released in our body by bacteria. Sorry. These antitoxins are used for injections. Okay, they are obtained from the blood of horses or other animals here. Now see the process here. The toxin producing bacteria, they are produced in the labs. Okay, then they are injected into the bloodstream of an animal here. The horse you can see there. These stimulates the formation of antitoxin. Now, when they enter the bloodstream of that animal, they stimulates the formation of antitoxins. Blood is then drawn from the animal. Okay, then the serum portion, which contains the antitoxin, is separated for injection. Okay, so always remember this antitoxin, they are allergic. Okay, humans, we are allergic to animal serum, so therefore it is repeated injection of this serum from the same species of animals are dangerous also. So presently, in general, the term antibody is used instead of antitoxin. Remember that. Okay, now you know what are antibodies. See here, antibodies are special protein molecules found in the blood which act against the germs or their secretion. Antibodies belong to a category. You can see the picture, the antibodies, antigens, the germs there, no pathogen, you can see there. So antibodies they belong to a category of immunoglobulin and is classified as protein. They are prepared or they are produced by the plasma cell in response to the antigens that are produced by the germs. You can see a bacteria is there, no, and you can see the bacteria is producing antigen. So when these antigens are produced by the bacteria, the plasma cell, okay, it contains the lymphocytes, okay, and this lymphocytes it produces the antibodies. Okay, this reaction, the production of the antigen and the antibody, is known as antigen antibody reaction. Then these lymphocytes, they are produced in the bone marrow. Okay, in infants they are known as B lymphocytes. These are white blood cells with a single large nucleus, okay, which is found mostly in the blood, lymph nodes, and the spleen. Okay, so in our body, um, can make unlimited varieties of different antibodies against the antigen produced by the invading germs. It depends upon different varieties. Okay, antibodies are also of different varieties according to the antigen that a germ, when it enters into our body, it produces. According to that, there are various types. They can produce varieties of antibodies. Antibodies, they are specific in nature. That means one type of antibody can act only on one particular type of antigen. Okay, an antibody, it recognizes a particular antigen that binds and it tries to make it harmless. Okay, it neutralizes it. Then some people, they are born with a particular antibodies present in their blood, such as people who do not suffer from that particular disease even if the germ have entered into our body. Okay, those, they have that antibodies present in their blood. That is why when the germs enter, the disease causing germs enter into your body, he is not affected by it. So immunity provided, okay, that are provided by the antibodies may be of short period and long period. Short period, they're passive immunity, and long period, they are active immunity. Okay, so smallpox, measles, and all, once the antibody is given to you all, it lasts for a longer time. No, and you can take an example of cold. You take antibiotics, medicine, you take it, so next year again you get it. Yes or no? Those are passive immunity, only for a short period of time. Hope you understood it. Here, to make you clear what are serum here. Okay, so serum, you can see, for you to make it clear, it is a yellowish fluid, clear yellowish fluid obtained, which is obtained after separating the blood. So when you take a blood sample, you keep it for a while, yes or no? So then what happens? The blood portion and the serum gets separated. Okay, the solid component and the liquid component, it gets separated. Okay, this is called blood serum. This blood serum from the tissues of immunized animal contains antibodies, okay, and are used to transfer immunity to other individual. So the serum, it contains antibodies that helps you to get immunized with different types of diseases. Okay, so this can be transferred. Okay, this can be helped, this can be used to transfer the immunity to another individual. So this is how antibodies are also formed. Okay, this serum, it contains the antibodies. We take those antibodies, and those antibodies, if it is capable of fighting the germs that has entered, okay, those are used as vaccines also. Okay, yes, now antibiotics. What antibiotics? These are antimicrobial class of antimicrobials which are relatively harmless to the host and therefore can be used to treat infections. They are harmless, they just treat the, helps to kill the germs, neutralize the germs. Okay, and it is harmless for the host. So antibiotics is a drug produced by certain microorganism that has the ability to kill other group of disease causing organism. They are widely used in medicines because many of them kill or prevent the growth of disease causing bacteria, viruses, as well as fungi. I think so you have heard about the first antibiotic that was produced. So you should remember the name. It is penicillin. Okay, penicillin notatum, and it was discovered by Alexander Fleming. Okay, so penicillin was discovered okay, from a fungus known as Penicillium notatum. This penicillin or the penicillin, they are active against infection caused by bacteria, especially cocci bacteria, or cocci we can call it. Okay, there are various forms according to the shape and size. Okay, you'll study in the later classes. So the penicillin, it helps to treat the bacterial infection. You should remember that. Okay, it also includes it treats infections, bone infections, pneumonia, even sore throats and all, boils, and also certain cases of blood poisoning and all. Okay, so these are the benefits of penicillin. So antibiotics, they are used as medicines to fight infections, both in animals and humans. It is also used as a preservative. Okay, it helps in the preservation of fish and meat, fresh meats. And then this antibiotics, they are added to the animal's food to prevent internal infections. Okay, you can see here. What do you think these are? These are disinfectants. Okay, disinfectants is they are mainly used in hospitals, dental surgeries, kitchens, and bathroom to kill infectious organism. Okay, some of the famous important disinfectants, not famous, I'm sorry, but important disinfectants are chlorine. Okay, it is used in drinking water to bleach. Okay, the microorganisms that are present in the water can be killed by using chlorine. So you all know, I think so you have studied, no? Chlorine is a bleaching agent also. Then comes chlorine dioxide. Okay, it is used as an advanced disinfectant in drinking water to purify drinking water. Alcohol. Okay, so used over bruises on the skins and allowed to evaporate for quick disinfection. Then some hydrogen peroxide. They are used in hospitals to disinfect surfaces. Then ozone, the gas added to water for sanitation. Ozone. Okay. Then phenyl. Uh, these are used as household disinfectants. Okay. This phenyl is probably the oldest disinfectant, I guess, and was called as carbolic acid in the early days. Okay, it was known as carbolic acid. Now we call them phenyl. Another one is potassium permanganate, used to disinfect utensils and also glasswares in the laboratories. Sodium hypochlorite, used in homes to disinfect drains and toilets. So these are all the different types of disinfectants. They are used in non-living objects, remember that. Okay, and it is a chemical substance that destroys the germs. It sterilizes the non-living objects. Here you can see Dettol, Savlon. What are they? They are antiseptic. Antiseptic is a mild chemical substances that are applied on the external surface of the body, okay, which helps to kill or prevent the growth and the reproduction of the disease causing organisms. They reduce the possibility of infection of the germs. Okay, so some examples of antiseptic, you see Dettol, Savlon, no, boric acid, alcohol, hydrogen peroxide, also iodine, compounds of phenyl, sodium chloride, okay, benzoic acid. Then these are the mild chemical substances. Okay, they are available in many commercial products also. Okay, and they help to reduce the possibility of infections that may be caused by the microorganisms. So you should know the difference between antiseptic, disinfectants, antibiotics and all. You should be able to differentiate between it. Now comes your sulfonamides. Okay, these are groups of chemical antibacterial compounds only. They are the first drugs, okay, to be proved to safe and effective against the bacterial infections. They play an important role in antibacterial treatment. Okay, since the World War II, these drugs helped to save many lives during this Second World War. Okay, they are often given in a combination with antibiotics. Okay, because their prolonged use leads to the development of resistant strain of the bacteria. They are taken orally. Okay, and it is the safest and the easiest method, okay, of administrating drugs. This There are many sulfur drugs. Okay, the sulfur drugs may also cause, sorry, should be when you take this sulfur drugs and all, they should be administered under the supervision of the physician, because many people, they may be allergic to sulfur drugs. Okay, most serious complication of treatment is that it damages the urinary tract, so you should consult the physician. Okay, so there are other sulfonamides like um, they prevent the bacteria from multiplying. Okay, so sulfadizine and sulfonamide, okay, they are the groups or the compounds, okay, that helps to kill. It does not actually kills, but it prevents the growth of bacteria or the multiplication of the bacteria in a body. Okay, so for example, dengue, you have heard about dengue, no? So the sulfonamides are used. Okay, here the treatment of the dengue is done with a combination of antibiotic with one of the sulfur drugs. Okay, combination of streptomycin antibiotic, and along with that sulfur drugs are given. So it is always used with an antibiotic. So now at last you can see this picture is just for you all to have a glance how antibodies are produced. First, taken this one, antivenom. How antivenoms are produced? You know when you are bitten by a snake, what is given? Antivenom are given to you all. Okay, poisonous snakes. So how are these antivenom produced? Now see here. Snake. We take the snake, collect the venom from the snake. Then that venom is introduced into the animal, any animal, mainly the horse there, injected. Then what do you see? After that, after the animal get immunized, we take the blood of that animal, we take it, then we separated it. Okay, you know when separation is done, the blood gets clot and the serum, it gets separated. We take the serum. Okay, so in the serum we need to separate the antibodies, the Y shaped, you can see there, no, those are the antibodies. Remember, always remember, antibodies are Y shaped because it has to uh, what to say, to distinct path there, you can see there, no, Y shaped, which can cling or which can attach to the antigens. Okay, and it makes those antigen or the microorganism harmless. Then the antibodies are separated, and these antibodies are formulated and introduced as vaccines for particular treatment. Okay, So this is how antibodies are produced, how vaccines are produced. This is just an example to see you all, make you all clear. Hope you have understood and enjoy it.